Hi, my name is Cassie Starnes, and I'll be talking about Niffler population genetics. Now, this particular population of Nifflers has three different phenotypes. The genotype for the black-furred Niffler is, is Big B, Big B. The phenotype of brown and white has a genotype of Big B, Little b. The Little b genotype has a phenotype of the blue fur. Now, to assume this is Hardy-Weinberg, we must first assume that it's infinitely large. There's random mating occurring, no selection, no migration, and no mutations in this population. Now, these are the frequencies of each phenotype given by population studies. We know there's 37 black nifflers, 42 brown and white, and 39 blue, for a total of 118. To find the allele frequencies, we'll take the homozygote and multiply that by 1 and the heterozygote by 0.5. So for the frequency of the Big B allele, we'll do 37 times 1 plus 1/2 of 42, all divided by 118 for the total. This number is represented by P and is given as 0.4915. We'll do the same for the frequency of the little b allele, giving us Q, which is 0.5085. Now we'll insert these into our Hardy-Weinberg equation, which is P squared Big B plus 2PQ Big B little b plus Q squared. The genotypic frequencies are very simple. We'll take the black nifflers, which are 37, divided by 118, giving us the frequency of Big B genotype. We'll do the same for each genotype, giving us all of these numbers. Now, something important is that these numbers together should equal 1, because these are essentially the percentages of the population represented by each genotype. Now, these nifflers live in a particularly cheery forest until the Dementors came, killed everything. Forest became very dark, and it's no longer suitable for the brown and white nifflers. Now, this is disruptive selection because the homozygotes are both favored over the heterozygote. Something interesting is that the black nifflers camouflage slightly better than the dark blue nifflers, giving their fitness a slightly higher number. However, both of the homozygotes are favored above the heterozygote. The genotypic frequency after selection is shown here. We'll take the initial frequency times the fitness for each genotype, giving us the unadjusted survivors. To adjust this to the population, we'll add that number and divide each of them by the total, which is given down here as 0.6742, giving us the relative frequency after selection. The allele frequencies after selection are similar to what we did earlier. We'll multiply 1 times the Big B plus 1/2 of the heterozygote, giving us each of these numbers. These are my sources. Thank you.